This conference will now be recorded.
Good morning, all of you.
Good morning, ma'am.
Morning, ma'am.
Yeah. So today we'll start with terpenoids. Let me brief terpenoids, which was discussed in the last class, and then we will go on to other metabolites. According to your syllabus, there is triterpenoid in detail, and they have given it as other terpenoids and volatile oil. So for all these three things, that is triterpenoids, other terpenoids, and volatile oil, this becomes the basis. Okay. So as we had discussed in last class, terpenoids are made up of isoprene units. So a chemist by name Leopold Ruzicka, who was born in 1887, he showed that many compounds are found in nature and they had multiples of 5 carbons which were around arranged in the same pattern as that of isoprene molecule. And so he called them as terpenes, or hemiterpene, or simply terpene. You can see in the slide, head to tail condensation will be there. It takes a chair formation. So this is one isoprene unit which has 5 carbon atoms. Even if you see natural rubber is a polyterpene. It has similar units of isoprene joined together. So it has C5H8 taken. Let's come into the biological isoprene unit. Isoprene units in terpenes do not come from isoprene, they come from isopentenyl pyrophosphate. So we have discussed about acetate pathway or the mevalonic acid pathway, and know that acetate through mevalonate pathway it will form isopentenyl pyrophosphate, which has 5 carbons. Isopentenyl pyrophosphate has 5 carbons, and remaining will be multiples of these are nothing but natural products related to isoprene. So this is the structure of isoprene. It is nothing but 2-methyl-1,3-butadiene. Okay, if you look at the structure, it has 2 double bonds, and in which there are 5 carbon atoms and 8 molecules of acetic acid. Mevalonic acid, wherein mevalonic acid further gives rise to isopentenyl pyrophosphate. This is what we have studied in mevalonic acid pathway. Isopentenyl pyrophosphate, this is the structure. At the terminal end, it is having this OPP group. Another C5 units gets combined to this, wherein the PP unit is removed, and to the O molecule, other C5 units gets combined to form dimethyl allyl pyrophosphate or farnesyl pyrophosphate and other terpenoids. And this is also very clear to you, terpenes, and what is the meaning of terpene and what is terpenoid. Terpenoids are nothing but oxygenated derivatives of terpene. So apart from carbon and hydrogen, they will also have oxygen. So next is isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. These two are interconvertible, except there is a change in the double bond. So dimethyl allyl pyrophosphate has a leaving group, that is pyrophosphate, at an alicyclic carbon. So it is reactive toward nucleophilic substitution at this position. So if nucleophilic substitution occurs, this PP pyrophosphate group will react. And the carbon-carbon bond formation, so we have discussed about this also. The key process, it involves the double bond of isopentenyl pyrophosphate acting as a nucleophile towards the alicyclic carbon of dimethyl allyl pyrophosphate. So this will combine and a carbon-carbon bond is formed. And so here what happens, where you are seeing the plus group, carbocation is formed, and this can lose a protein to give a double bond. So you can see here isopentenyl or dimethyl allyl pyrophosphate, it will lose one carbon atom, it forms a carbocation, it will lose a proton. So it will lose a proton to give a double bond. And that is how the next structure, geranyl pyrophosphate, occurs. So it can undergo hydrolysis of pyrophosphate to give geraniol, and this geraniol is one of the examples which is present in rose oil. So here see, after carbon-carbon bond formation and hydrolysis, isopentenyl pyrophosphate will form geranyl pyrophosphate, that is, and then geraniol. So geraniol pyrophosphate will undergo hydrolysis. You can just see over in the slide. What happens here is the pyrophosphate PP group is removed. This PP group is removed, and it takes up geraniol. So geraniol is nothing but a constituent present in a terpenoid present in rose oil. Now next, from 10 carbon, how it forms 15 carbon. So to 10 carbon, geraniol pyrophosphate has 10 carbon atoms. To this, one isopentenyl pyrophosphate which has 5 carbon atoms will combine. So at this double bond point, it will combine. So carbocation is formed there, and then it will form C15 units. So you'll get this particular compound. This is nothing but farnesyl pyrophosphate. So here, wherever carbocation is there double bond occurs, and this compound is called as farnesyl pyrophosphate. So hydrolysis of this pyrophosphate ester, again here pyrophosphate PP is removed, and from this PP OH it is replaced by H, and we get this alcohol called as farnesol. Next, sometimes it undergoes cyclization by intramolecular carbon-carbon bond formation, and it gives various isomers. Okay, is it clear till here?
Yes, ma'am.
Fine. Next, we discussed about classification of terpenes. So now if we talk about classification of volatile oils, so we'll just finish off terpenoids. After that, we'll come back to volatile oil, which almost has the same type of classification also. But there is another classification of volatile based on the functional group it has. Okay, like alcohol, if it has OH group, it is called as alcohol volatile oil. If it has CHO group, it is called as, if CHO group is
Aldehyde.
present, it is called as aldehyde volatile oil. Good. If it has a CO group, then it is called as, if CO group is present, what is it called?
Ketone.
Ketone volatile oil. So like this, based on the functional group, again volatile oil is also classified. And now this classification, whatever you're seeing on the slide, it is nothing but the classification based on the number of terpene units. Clear, number of terpene units. I think this was clear in the last class. I'll just brief you here. Now, if it has C5H8 units, it is called as hemiterpene. What is the other name for hemiterpene? What is the other name for hemiterpene?
Isoprene.
Very good. Isoterpene, or it's also called as isoprene. Okay, it is also called as isoprene. Yeah. Next thing is, if it has C10 units, that is C5H8 taken 2 times. So it becomes C10H16. Then it is called as
Monoterpenes.
it's called as monoterpene. Very good. Next thing is, if it has C5H8 taken 3 times, then it is called as C5H8 taken 3 times. Fast, who can answer this? C5H8 taken 3 times means what is its molecular formula?
C15H24.
So what is it called as?
Sesquiterpene.
Sesquiterpene. Very good. Next, if it has C5H8 taken 4 times.
Diterpenes.
Diterpene. Very good, Shashank. And how many carbon atoms diterpene has?
C20.
20. H32.
C20H32. Good. Next thing is, what we also studied if it has 5 isoprene unit, that is C5H8 taken 5 times. What is it called as? If somebody can remember, C5H8 taken 5 times. Don't remember? Can you recall sesquiterpene? Not sesqui, sesquiterpene. SESQUER. Yes?
Yes, ma'am.
So sesquiterpene has C5H8 taken 5 times. So see how many carbon atoms sesquiterpene has? Yes?
25.
25. So some things which are rarely occurring in nature, it is not considered in the main classification, but for your knowledge, you should know it. Next, diterpene. What is the formula for diterpene?
C20H.
That is a
32.
molecular formula. In terms of isoprene unit,
4.
yes,
C5H8 4
Bhagya.
times.
4
Okay, good.
isoprene.
Shashank, C5H8 taken
4 times.
5H8 taken 4 times as diterpene. Correct. Next, if C5H8 is taken 6 times, it is called as
Triterpenes.
triterpene. How many carbon it has?
30.
C13.
30 carbon. Okay, and how many hydrogen it has?
48.
Very good. Next, coming to tetraterpene. So it has C40. So what should be its formula in terms of isoprene units?
C5H8 taken 8 times.
5
8
H8
times.
taken 8 times.
C5H8 taken 8 times. So how many hydrogen it has?
64.
64.
64 hydrogen. Very good. Next, what is the formula for polyterpene?
C5H8
C5H8
taken
taken
1
N
number
times.
Taken n times. Very good. I hope all of you are clear with this, isn't it?
Yes, ma'am.
So this slide talks about various terpenes, whichever we have discussed now. So hemiterpenes means it has single isoprene unit. Okay, it has a single isoprene unit. Next, isoprene means hemiterpene. Okay. Next is monoterpene. It has 2 isoprene units with molecular formula C10H16. Examples. So now everything you know except for examples should get registered in your mind now. What are the examples of monoterpenes? What are the examples of monoterpenes?
Limonene,
Yes, it's there in the
geraniol,
slide itself. Geraniol,
terpeniol,
then next
limonene,
limonene,
terpeniol.
limonene, and terpeniol. Okay, geraniol, limonene, and terpeniol. Next, sesquiterpene. It is made up of 3 isoprene units. So its molecular formula C15H24. What are the examples of sesquiterpene?
Humulene, farnescence,
Humulene,
farnesol.
farnesene, and farnesol. Okay, humulene, farnesene, and farnesol. Next one, coming to diterpenes. So what does diterpene means? They are composed of 4 isoprene unit and they have the molecular formula C20H32. And from where are they derived? Yes, from where are they derived? From which pathway all these terpenoids are derived?
Mevalonic acid pathway.
Geranyl pyrophosphate.
Yes, it is derived from mevalonic acid. That is, it is from especially this diterpenes are derived from geranyl pyrophosphate. So in the previous slides, we saw how many units this geranyl pyrophosphate had. It had 10 carbon atoms. Okay, so geranyl means C10 plus C10, it becomes C20. Did you get it? Understood?
ma'am.
So examples are, you can remember taxidin and kefestol. Next, coming to ester terpenes, they have 25 carbon atoms, that is 5 isoprene unit, and they are derived from geranyl, geranyl farnesyl pyrophosphate. Okay, so geranyl has C10 units, farnesyl pyrophosphate has C15 units. So C10 plus C15 will give you C25 units, that is geranyl farnesol. Clear?
Yes, ma'am.
Yes, ma'am.
Next is triterpene means 6 isoprene units. So molecular formula C30H48. You have a linear triterpene. Linear means it does not form a cyclic chain. So linear means straight line. So example is squalene, which is a major constituent of shark liver oil. This is got by coupling two molecules of farnesyl pyrophosphate. So how many carbon atoms farnesyl pyrophosphate has? Yes,
15.
15, C15. So two molecules of farnesyl pyrophosphate will combine to get C20. Clear? So squalene is then processed biosynthetically to get lanostirol or cycloartenol, and this is the structural precursor for all steroids. Okay, if you remember the biosynthetic pathway, whatever we had discussed, from this farnesyl pyrophosphate we get squalene. Satyam, I think you can mute your mobile. So squalene is got from farnesyl pyrophosphate, and squalene will again combine with farnesyl pyrophosphate to give lanostirol. Lanostirol will further give rise to cholesterol, and then other steroids are obtained. Clear? Is it clear
Yes,
till
ma'am.
here? Next comes sesquiterpenes. So this is composed of 7 isoprene unit, and it has a molecular formula C35H56. They are microbial in their origin, means they are present in microorganism. Example is fergucadiol and tetraprenyl curcumin. Curcumin is got from this particular compound is got from curcuma longa, that is turmeric. Okay, next one is tetraterpenes, which has 8 isoprene units. So molecular formula C40H64. Biologically important tetraterpenes will include acyclic lycopenes. Lycopene is present in, last class I told you this. Where is lycopene present and where is carotene present?
Carotene is present in carrots.
Very good. Alpha carotene, beta carotene is present in carrot. What about lycopene? Anybody?
In tomato.
Good, Aishwarya. It is present in tomato. Next is polyterpenes. So they have long chains of many isoprene unit. Example is natural rubber. Then you also have something called as nor-isoprenoids. They are nothing but C13 nor-isoprenoids, and they are present in grapes, etc. Okay, will not go into the depth of nor-isoprenoids. Next, to generalize about terpenes, they are usually multiples of 5. They'll either have C5, C10, C15, C20, C25, so multiples of 5 units. And each group of 5 carbon atom is an isoprene subunit. Okay, so the building block of volatile oil is isoprene. Now they can be saturated or unsaturated, and many contains oxygen. So if it contains oxygen, terpenes are called as terpenoids. Clear?
Yes, ma'am.
Is it clear till here? Okay. Next, how does each isoprene unit join? We are telling they are multiples of isoprene unit. So they can join either by head to tail condensation or tail to tail condensation. So in this slide, very clearly the color is given and it is depicted here. So in the second case, where there is tail to tail condensation, you can look at the cyclic structure. Extra bond will be formed in between the two C5 units. and it will form a cyclic structure. Clear? Is it clear till here?
Yes, ma'am.
Yes, ma'am.
Okay. Next, coming to monoterpenes. So we have alpha-phenandrine, which is present in eucalyptus. Then menthol, very well known menthol. It is present in peppermint. Where have you heard of this word menthol? Have you heard of this word menthol?
Flavoring agent.
It uses flavoring agent in any commercial products. Have you heard or come across this menthol?
Toothpaste.
It is there in toothpaste, then
Mouthwash.
mouthwash, after shaving lotions. It has antibacterial effect as well as it gives a cooling effect. You can also see it in chewing gums, isn't it?
Yes, ma'am.
Yes, ma'am.
Okay. Next example here is a cyclic structure of citral, which is derived from lemongrass. So here you can see monoterpene means how many carbons should be there?
5, ma'am.
Monoterpene,
10.
10, ma'am.
10 carbon atoms. So you can see all these structures have 10 carbon atoms in them. Along with how many hydrogen atoms will be present? 16 hydrogen atoms along with oxygen. So it is called as a terpenoid. Clear? So very nicely you can see here. Each color represents one C5 unit. Okay, each color represents one C5 unit, and the yellow color represents the bonds. Okay, so if you see alpha philandrine, how many C5 units are there in it?
One. Two.
Two. One is pink color one, and one is a blue color one. So two C5 units are present. In menthol,
Two.
two again. In citral,
Two again.
two again. The yellow color ones are nothing but double bonds present in them. So see, without bonds, it will look like this. With bonds, it will be a complete structure. Clear? Next, coming to sesquiterpene. So sesquiterpene means how many C5 units it should have? 3 units. Good, Bhagya. So one example here is alpha selenine. This is present in celery. Okay. So how many units you can see here? How many C5 units you can see here?
3.
3,
3.
ma'am.
The pink one is 1 C5 unit, blue one is 1 C5 unit, and the green one is 1 C5 unit. All the yellow ones are the bonds added to it. Okay. Next, coming to diterpene. So now let me just ask you this way. Okay. If it is like this, okay, you have a yellow colored C5 unit, blue colored one, ash colored one, and green colored one. So bonds are formed here. So this will form which compound? Which class of terpenes rather? It will form
Diterpenes.
diterpene because how many C5 units it should have?
4.
4.
4 units. Clear? Next, yes. Just count how many isoprene units are there in this particular structure. There is tail to tail condensation of isoprene unit. How many units are there?
6.
6, ma'am.
So 6 means what should that class of terpenes be?
Triterpenes.
A triterpene. So best example is squalene, which is present in shark liver oil. And remember, we studied that it is a straight line. It's a linear structure, not a cyclic structure. Clear?
Yes, ma'am.
Next, this is farnesol. So how many units this farnesol has?
3 units.
3 units. See, 3 double bonds. So usually it is 3 units with a OH group present in it. And this farnesol is derived from
Farnesyl pyrophosphate.
very good. Farnesyl pyrophosphate via which pathway?
Mevalonic acid, ma'am.
Mevalonic acid pathway. So how do you get farnesyl pyrophosphate? When one isoprene combines with geranyl pyrophosphate, you will get farnesyl pyrophosphate. Clear?
Yes, ma'am.
How will you get geranyl pyrophosphate? When one isoprene unit combines with isopentenyl pyrophosphate, you will get geranyl pyrophosphate. Clear?
Yes, ma'am.
How will you get isopentenyl pyrophosphate? When acetate combines with acetic acid, you will get isopentenyl pyrophosphate. Clear?
Yes, ma'am.
So we have discussed the mevalonic acid pathway in the backward now. That's it. Okay. So this farnesol is used as perfume, it is used as pesticides, it is used as pheromones, anti-tumor agents, antibacterial drugs, etc. Okay, next this is the structure of chlorophyll. So chlorophyll A and chlorophyll B also, it undergoes tail-to-tail condensation. This is also derived from mevalonic acid pathway. Okay, see this is beta carotene. So how many C5 unit it has? You can look into the color of the C5 units and tell me how many unit it has.
8, ma'am.
It has got 8 units. So it is a tetraterpene. And if you look at the structure, so you have one cyclic ring remaining all linear structure. This is the structure of beta carotene, which is present in carrot. It is a best example for tetraterpene. Okay, it has 8 isoprene unit, 40 carbon atoms. And if you elaborate the structure, you can see a structure like this. Okay, next this is Taxol. So Taxol is again a terpenoid. It is obtained from Taxus brevifolia. It's a potent anti-cancer drug. Okay, next is camptothecin. This, okay. So till here we have discussed about terpenoids. Is there any doubt in terpenoids or volatile oil?
No, ma'am.
No, ma'am, no doubt.
No doubts. Okay, so okay, fine. Now just coming to the general characteristics of volatile oil. What is the, if I ask you the general nature of volatile oil, what all can you tell me?
It evaporates, ma'am.
Yes, come out with general characteristics. Very good. Volatile oil means, as the name suggests, it is volatile. It evaporates when left at room temperature. Next, any other general character you can think of?
Aromatic in nature. I mean,
When I
good
ask you,
fragrance.
yes, good. It is aromatic in nature. Any volatile oil has a odor. It has got a good smell. They are odorous principles of the components present in the plant. Okay, next. Next,
Degrades at high temperature.
its nature. Okay, it gets degraded at high temperatures. Good. Then what about its appearance, its look? What is its state of matter?
Liquid, clear liquid.
They are liquids, okay? They are most of the time liquids, but some examples are there, like camphor, for example, it's a solid, okay? So if you have to generalize, they are liquids. Then many a times colorless, but some have color. Then don't you think you have to talk about the solubility issue here?
Insoluble in water.
Very good. They are insoluble in water. So they must be soluble in what?
Alcohol.
They must be soluble in alcohol and other organic solvents. Very, very important. Only if you know this, you can remember the extraction and isolation procedures. So I hope it is clear till here.
Yes, ma'am.
Yes, ma'am.
Okay, next we'll go, if we see in your syllabus. So alkaloids we have completed. Then we are left with, phenyl propenoids and flavonoids. Now phenyl propenoids, okay, means They are nothing but phenolics. Generally, we also call it as phenolics. So these are derived from aromatic amino acids such as phenylalanine, tyrosine, and tryptophan. So through which pathway these will be derived? Phenolics. Yes. Aromatic amino acids means through which pathway these phenolics are derived. So starting material here is phenylalanine or tyrosine or tryptophan. So through which pathway? Yes, you can hear me?
Yes, ma'am.
Through which pathway these phenolics can be derived?
Shikimic acid pathway.
Very good. This is derived from shikimic acid pathway. Recall what we have discussed in previous classes. End product of all shikimic acid pathways, amino acids, and this amino acid will be the starting material for deriving lignans, flavonoids, coumarins, alkaloids, phenols, etc. Okay, so phenolics are also derived from aromatic amino acids like phenylalanine, tyrosine, and tryptophan, and all these will contain the structure which is derived from phenol. Some examples are, you have coumarin as an example here. Then these coumarins will have many antimicrobial agents, many feeding deterrents, germicides, inhibitors, etc. Next, lignin. You have heard of lignin, right? So lignin also comes under phenolics. You have they have given you as a drug in your syllabus, but then lignin is also a secondary metabolite. Okay, it is a secondary metabolite, and the plants which are rich in this particular lignin is phyllanthus. Phyllanthus neruri you have, and there are some plants which are rich in lignin. So you have got to study those drugs under this particular chapter. Fine. So lignins which are abundant in secondary cell wall, they are rigid and they are resistant to extraction, or many times they get degraded to anthocyanins, flavonols, etc. And phenols are present in every plant. They attract pollinators to the plant. Okay. So what you should know from this slide is phenolics. They are mainly derived from phenylamine, phenylalanine, tyrosine, and tryptophan, and these all have the structure which is derived from phenol. So they can be classified into coumarins and lignins, which where lignin is further classified into anthocyanins, flavones, and flavonols. So all these come under phenolics. Okay, flavonoids means anything which gives color to the plant usually is a flavonoid. Is the brief introduction to phenyl propanoids clear?
Yes,
Yes?
ma'am.
Yes, ma'am.
So, yeah. So lignins and flavonoids, they come under this phenolics. Even phenyl propanoids, these are nothing but the chemical structures present. Okay. And under that, in your syllabus, you've got to study the drugs like lignans, T, and ruta. Okay. Next is glycosides. In glycosides, in general, they have given cardiac glycosides, and they have also given steroids. So steroids can be steroidal alkaloid as well as steroidal glycosides. Now, steroidal alkaloid we have studied while we are discussing about alkaloids. Steroidal glycosides, we'll be studying it under glycosides. Okay. Now, what are glycosides? Yes. What are glycosides made up of? Usually they have two moieties, isn't it? What are they?
Sugar and non-sugar moieties, ma'am.
Yes, a sugar and a non-sugar moiety. That is called as glycone and aglycone. Okay, glycone and aglycone. What is the sugar moiety in this? Glycone or aglycone? Which is the sugar moiety?
Glycone. Glycone.
Glycone is the sugar moiety, and aglycone is the and aglycone is the non-sugar moiety. And which one gives pharmacological action to the plant?
Aglycone.
What do you think whether glycone gives the pharmacological, yes, A-glycon gives a pharmacological action. Okay. So glycosides are compounds or they are secondary metabolites which on hydrolysis yield a glycone and a no and a A-glycone moiety. A-glycone moiety will be having the pharmacological action, whereas glycone moiety or the sugar is mainly meant for transportation of A-glycone to the required site. Clear? Transportation of A-glycone to the required site. Okay, and usually these glycosides are present in vacuoles or they are in inactive form. And the glucose enolates, they are found primarily in mustard family. They give the pungent taste. Now, this glycone and glycone, basically they are linked by certain things. They can be linked by carbon atom, oxygen atom, nitrogen atom, or sulfur atom. So, one type of classification of glycoside is based on the linkage of the glycoside bond. So you have C glycosides, O glycoside, N glycoside, and S glycoside. So in Kokate, a very nice classification is given. Pallavi, you click the classification of glycosides and you send it after the class.
Yeah, ma'am, sure.
Pallavi is there in the class?
Yes, ma'am.
Yes or no? I
Yes,
can see you
ma'am.
here, but you're not answering.
I'm there in the class.
You click the
Actually, she
next
is talking.
Okay. So based on the linkage, we have seven. Next comes resins. So usually they occur as latex, okay, or the milky fluids. And phytosterols or steroids, this encompasses sterols. They are steroidal compounds which are similar to cholesterol. They occur in plants and they have carbon side chains with or without a double bond. Okay. Next, you have different type of glycosides like anthracene glycosides, digitalis. You have digitalis glycosides, that is cardiac glycosides. Sorry, cardiac glycosides. Then you have cyanogenetic glycosides. Then you have flavonoid glycosides. Okay. Then you have isothiocyanate glycosides, steroidal glycosides. So these are the different types of glycosides based on the functional group present. Okay, then we were discussing about essential oil. So essential oil is nothing but it is something which has aroma. Volatile oil only is called as essential oil. Essential oil because it forms the essence of the plant. Essence means main heart of the plant. It is also called as ethereal oils because ether means, all of you know that ether means what? Ether means nothing. Okay, if you leave ether to room temperature, what will happen? Yes, what happens if ether bottle you open and keep it at room temperature?
Evaporates.
It evaporates and leaves behind. It evaporates and leaves behind. What does it leave behind after evaporation? See, if ether bottle is there, if you open it, what will happen? You can hear me?
Yes, ma'am.
Am I audible? What happens if you open ether bottle? What is left behind in the bottle?
Nothing.
Nothing. So similarly, volatile oil, if you open it and leave it at room temperature, you're left with nothing. That's why they are called as ethereal oils, or it's also called as A-ether olia. Okay, and basically these volatile oils are used in perfumes, cosmetics, soaps, and other products for flavoring food and drinks, etc., and for adding scents to incense stick, that is agarbatti and all, and for household cleaning products. So anything with good smell has volatile oil. So phenyl propenoids, we have discussed, these are nothing but diverse family of organic compounds. They are synthesized by plants from amino acid phenylalanine. They are found throughout the plant kingdom. They are the essential component of a number of structural polymers. They provide protection from ultraviolet light. They defend against herbivores and pathogens, and also they help in plant pollinator interactions as floral pigments and scent compounds. This we have discussed already. Okay. So these are some of the secondary metabolites. Besides that, you have resins, tannins, iridoids, naphthoquinolines. These iridoids and naphthoquinoline, they come under the main classifications of other secondary metabolites. Whereas if you look into um, tannins, so what are tannins? What is the general nature of tannin? Yes. What is the general nature of tannin? What taste it has, or what is the use of tannins? No answer?
Astringent.
Very good. They are astringents. Astringent means what? If you eat amla or if you eat pomegranate, you have a sort of sensation in your tongue, isn't it? That is called as astringency. And if you look at the uses, astringent means it will precipitate proteins. Okay, astringent means it will precipitate proteins. So tannins, they are the most widely occurring secondary metabolites. They are present in solution form in cell sap and also in distinct vacuoles. Example is black catechu, pale catechu, myrobalan. So last year's pharmacognosy, if you have gone through these metabolites, you will know its exact definitions. And glycosides, yes, we discussed that they are a wide which has a glycone aglycone portion. And resins are amorphous plant exudates of complex chemical nature. So they occur as solids or semi-solids. Basically, they are amorphous mixtures and they contain essential oils, oxygenated products of terpenes, that is terpenoids and carboxylic acid. Usually, terpenes are the Can you hear me?
Yes, ma'am, but can you repeat the previous sentences?
From resins, you want me to repeat? Fine.
Yes,
So
ma'am.
resins are amorphous plant exudates. They have complex chemical nature. They can occur as solids or semi-solids. Basically, they are amorphous mixtures of essential oils, then oxygenated product of terpene, that is terpenoids, and carboxylic acid. Means what? Chemically, they contain volatile oils, terpenoids, and carboxylic acids. They are usually the end product of terpene metabolism and they are formed within schizolysigenous or schizogenous ducts or cavities which are present either internally or externally. So example is ginger, oleoresin, capsicum resin, etc. Now what is this schizogenous or schizolysigenous? Just imagine there are two cells and there is a split in between. So in between a duct will be formed and it will produce this resin. This is called as schizogenous duct or schizolysigenous duct. Schizo means split. When there's a split between two cells, a gland will be formed and within this gland these resins will be stored. Yeah.
Yes, ma'am.
Is it clear to you? Okay. So now I have actually covered the definitions and the sources of and some uses of all the secondary metabolites. Now your task begins here. So for assignment, I'll be giving you one or two drugs, or I can give you one chemical class and one drug. If you get chemical class, you have got to write about its introduction, its composition, chemistry, chemical class, biological source, therapeutic use, and commercial application as given in your syllabus. Based on those guidelines, you can give. And for the drugs, I have made a separate PPT, like in what slide what information has to be given. So follow that particular slide. Information you can draw from Kokate, and then additional pictures, additional information you can take from any books or from the internet also. Okay, try to make your presentation as good, as interesting as possible. Clear? And in case you want to give it on Zoom, you can even use the live samples. For example, Vinca, always you can get the flower, you can get the plant, you can get the leaf. Then tea you can make it more interesting. Okay, you can end your presentation by having a cup of tea. So it is left to you as how interesting you're going to make it. And this is the assignment which is going to be given to you, and this is going to fetch marks. Is it clear? So by today or tomorrow, I'll just list out the names and make and give it to you. One by one, you can start presenting. Clear?
Yes, ma'am.
So I think one or two drugs I will present and show you. Maybe in tomorrow's class, I will present and show you how you have got to present a particular drug. Okay. So based on that, you can start preparing the presentation. I don't want any absentees. So hardly 30 drugs plus 8 constituents. So each one of you may get either 2 drugs or 1 chemical class and 1 drug. Clear?
Yes, ma'am.
Okay. Now the next thing is, what about your test? Today we were supposed to take a test. Everyone, are you ready? Will you write it on tomorrow or on Wednesday?
Yes, ma'am.
When? Tomorrow or Wednesday? When you want to write? When you want to write?
Ma'am, Wednesday we
Tomorrow,
have one
ma'am.
more test.
Tomorrow or Wednesday? When?
Tomorrow, ma'am.
Ma'am, Wednesday we
Mahandesh,
have
just take the majority and tell. Wednesday you have?
Medchem, ma'am. Shilpa ma'am's test is there. So tomorrow only we'll write.
Tomorrow you want to write. Fine, no problem. Okay, so tomorrow be ready for test on biosynthetic pathways and tracer techniques. The theory part as well as the biosynthetic pathways. Any doubts in today's class?
No, ma'am.
Just a small extraction process was remaining, isn't it, for alkaloids? So I think Pallavi has shared with you all in the group. So main basis for isolating alkaloids is its basic nature and its solubility pattern. We know that alkaloids can exist in the form of free bases as well as their solubility pattern. We know that they are soluble as free bases in organic solvents. As salts, they are soluble in water. So this is considered, and a method is designed. That method is called as Stas-Otto method. STAS-OTTO, Stas-Otto method. So even in my notes, you can see the flow chart of it. So here basically moistened drug is taken and to this alkali is added. So already we know bases, alkaloids are bases. And if you add more alkali, what will happen? Free base will be set out. Okay. And this free base can be separated out with polar solvents. Okay. So alkaloids are transferred to polar solvents. Now evaporate the alcohol layer. You'll get the extract, add organic solvent to this. Then you take out the aqueous phase and make it alkaline. Okay. So slowly you just go through the flow chart one by one based on solubility pattern. You'll be able to understand. You're going to make it alkaline with either sodium carbonate or with ammonia. Then you're going to extract with suitable organic solvent, filter and evaporate. So this is one method. Another method is you can take the crude drug and add ammonia to this. So this will convert all alkaloidal salts into free bases. Now free bases, you know that they are soluble in organic solvents. So you'll add solvent like ether, benzene, chloroform, etc., where you get free alkaloids. Now this is further purified by crystallization, steam distillation, chromatography, gradient pH technique, fractional liberation, etc. Let me just tell you the second method once again. You take the drug and you add ammonia. What will happen? All the alkaloidal salt will become free bases. Now free bases, you're going to extract this free base with, we know that it is highly soluble in organic solvent. So you either extract it with ether, benzene or chloroform. You're going to get alkaloids. This alkaloid you will further purify by crystallization, steam distillation, fractional liberation, chromatography or gradient pH technique. If it is salt, what you'll do? You'll take out the salt and you'll add polar solvent to this. So it comes out into polar solvent, and then you'll evaporate this and add alcohol, extract again with organic solvent. So the free bases would be set aside. Then you're going to make it alkaline with either sodium carbonate or ammonia. So either of the method. Nothing, you should just remember, if you know its basicity, if you know its basicity pattern, and if you know its solubility pattern, you can easily extract the drug. Nothing, you should make it basic. If you make it more basic, you're going to treat it with organic solvent. If you're going to convert it in the salt form, you're going to extract with water and then again set the free bases by treating it with organic solvent. Clear?
Yes, ma'am.
Okay, so I'll stop the class here. So we'll continue the remaining in the next class. Bye bye. Have a nice day.
Thank you, ma'am.